Hello, everyone. Welcome to the Emerging Trends in Pharmaceutical and Biopharmaceutical Analysis. I'm Jerome Workman, the Executive Editor of Spectroscopy, and I'll be your moderator for this event. We are pleased to bring you this afternoon session, which is part of a two-day virtual symposium presented by Spectroscopy, the Society for Applied Spectroscopy, and the Koblenz Society. The Society for Applied Spectroscopy, or SAS, is a nonprofit organization dedicated to the dissemination of information related to spectroscopy. SAS is pleased to be the publisher of the internationally recognized peer-reviewed journal Applied Spectroscopy. The Koblenz Society is a nonprofit organization whose purpose is to foster the understanding and application of vibrational spectroscopy. Both societies have an educational mission, and this event is part of that mission. We invite you to visit both societies' websites to learn more. This virtual symposium is sponsored by CEM, PerkinElmer, Thermo Fisher, and Metrome, and Endress and Hauser. And now a statement from our sponsors. Today's event would not be possible without the support of our sponsors. CEM Corporation began manufacturing analytical instruments in 1978. In 1985, CEM developed the first microwave digestion system, revolutionizing sample preparation for trace metals analysis. Since then, continuous innovation has kept CEM at the forefront of sample preparation. With worldwide headquarters located just outside of Charlotte, North Carolina, CEM operates 7 subsidiaries as well as a large dealer network covering the globe. CEM truly is the global leader in microwave sample preparation. At Metrome USA, we're more than a leader in chemical analysis. We're a community driven by passion, integrity, and a commitment to quality and innovation. With expertise in both laboratory and process systems for titration, ion chromatography, electrochemistry, and spectroscopy, our approach goes far beyond just technology. Discover how our dedication to both science and humanity shapes everything we do at www.metrome.com. Customers worldwide trust Thermo Fisher tools and solutions to help them accelerate innovation and enhance productivity. Together, they are making advancements that truly matter. Thermo Fisher achieves this by providing an unmatched combination of innovative technologies, purchasing convenience, and comprehensive support. PerkinElmer is a global provider of scientific solutions with an 80-plus year history of bringing expertise, innovation, and technology to our customers. They manufacture the latest in analytical instrumentation and provide essential lab support services to streamline workflows. Partnering with scientists, clinicians, and lab technicians, they accelerate scientific outcomes and aid the development of groundbreaking products. PerkinElmer believes in the power of science towards a healthier, safer, and more sustainable world. Endress and Hauser provides advanced measurement and automation solutions for industrial processes across the globe. Their offerings include instruments for flow, level, pressure, temperature, and liquid and gas analysis. With a focus on innovation, reliability, and digital integration, they serve a plethora of industries. Their portfolio comprises of many products for any application, from basic to highly specialized, ensuring efficient and sustainable process control. We have a few important announcements before we begin. This symposium is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found at the bottom of your media player. Please note that all questions will be answered offline after today's session. So when you type in your question, please include the name of the speaker that your question is for. You can enlarge the slide window by clicking on the icon in the lower right-hand corner of the video player. The slides will advance automatically during the event. If you have any technical problems viewing or hearing this presentation, please click on the question mark help widget located at the top right of your presentation window. I would now like to introduce our speaker, Nicole Kubler. Nicole Kubler is the National Business Development Manager for Raman at Andreessen Hauser, where she leads strategic initiatives to expand market presence and drive growth in Raman spectroscopy solutions. With a Master of Engineering in Aerospace Engineering and a Bachelor's in Mechanical and Aerospace Engineering, Nicole brings deep technical expertise and a passion for innovation. She excels in cross-functional collaboration, data-driven strategy, and customer engagement. Nicole is dedicated to delivering high-impact solutions that optimize performance and efficiency across industries, positioning Andreessen Hauser as a leader in advanced analytical technologies. Nicole, thank you for joining us today. Please get us started.
Hi, everyone. Thank you for tuning into our Raman session. My name is Nicole Kubler, and today we will focus on the application of Raman spectroscopy in various processes and explore how Raman spectroscopy can be utilized to enhance the accuracy and efficiency of material identification, quality control, and process monitoring. Starting off with our timeline, Andreessen Hauser has over 40 years of experience in optical analysis, where Andreessen Hauser acquired Kaiser Optical Systems, the global leader in Raman spectroscopy, in 2013. Kaiser Optical Systems Inc. was founded in 1979, developing state-of-the-art holographic technology, where Kaiser brought expertise in solid, liquid, and gas analysis. After this acquisition, the formation of Andreessen Hauser Optical Analysis was established in January 2022. So what is Raman spectroscopy, and how does it work? Raman spectroscopy is a type of non-destructive analytical technique that provides molecular and chemical information based on the scattering of photons by molecular vibrations. So what happens on a molecular scale? When a laser source interacts with a sample, energy is delivered in the form of photons. This interaction produces Raman scattering, where the energy of scattered photons shifts slightly. This change is known as the Raman shift. These shifts can be measured across a spectrum of possible energy changes, creating a unique molecular fingerprint. Every molecule responds differently when stimulated by light. For example, glucose, lactate, fructose, galactose, each absorb and scatter light at different wavelengths and intensities, allowing us to differentiate them. What the animation shows, the laser light travels through one fiber optic path to the sample, and the scattered light is collected through a second fiber optic path. From there, it passes through a series of filters, gratings, slits, and mirrors before reaching the detector or camera. This process generates the Raman spectrum, which displays discrete peaks corresponding to the molecular vibrations of the sample. Raman can provide three types of analysis. The first being qualitative. By analyzing the Raman spectra, specific peaks corresponding to the product molecule can be identified. The second is quantitative. Raman spectroscopy can provide a quantitative measure of the product concentration by correlating the intensity of Raman peaks associated with the product molecule. In complex mixtures, we use multivariate analysis to unmix the fingerprints that are present in an acquired spectrum. These Raman shift spectra can be correlated using techniques like partial least squares for regression or other regression techniques to changes in constituents or changes to other attributes of the process media, where a calibration curve or model can be established to estimate the product concentration. For example, changes in the concentration of glucose, in lactate, amino acids, or even VCD over the course of the process. In this way, Raman spectroscopy offers real-time process monitoring and even automated process control. By adding an additional axes of time, we are now able to track how and when a reaction or batch is completed, which is crucial data for optimizing process conditions, assessing productivity, and ensuring product quality. So the benefits of Raman is monitoring of those critical process parameters. Raman can monitor nutrient levels, metabolite concentrations, and cell viability in real time. This allows for precise control over process conditions, minimizing batch-to-batch variability, and leading to more predictable manufacturing outcomes and repeatability, as well as real-time process analytics, allowing for real immediate detections of deviations from optimal conditions, leading to more efficient use of resources and higher yield per batch. All of these provide significant reductions in production times and increased overall productivity, reducing time to market. Now we're going to dive into the hardware, starting with our analyzers. On this slide is the RxN2 on the left and RxN4 on the middle. Although different in appearance, they have the same functions and internal modules that allows for lab-to-process transferability. These instruments have one laser source, which can be configured at 532 nanometers, 785 nanometers, or 993 nanometer excitation wavelengths. The choice of wavelength of the laser source depends on the required application. Raman spectroscopy is most often performed using laser sources at a wavelength of 785 nanometers. This wavelength source is often selected as it balances the competing factors between Raman signal intensity, fluorescence, and detector sensitivity. Both instruments can be configured as either a single channel or a 4-channel system, where again the differences lie in the outer case or enclosure and orientation of the instrument. The RxN2 on the left is designed for lab and pilot scales, where it can be mounted on a cart or benchtop for location flexibility. And the RxN4 in the middle is designed for use in process and manufacturing environments, which could be either rack-mounted or in a NEMA 4X enclosure. The analyzer on the right is our RxN5, our gas analyzer, where the key difference between this instrument and the RxN2 and 4 is that it can be configured with multiple lasers, allowing for simultaneous measurements in a gas environment. This system can only be configured with a 532 nanometer wavelength. All of our probes are uniquely designed to enable non-invasive compositional analysis of material without needing to extract, prepare, or destroy the native sample, or even modify any existing hardware. Each probe line is engineered with specific windows, materials, and sampling features tailored to its intended application, allowing for environmental flexibility for any phase of material, whether the application is solids, liquids, gases, or turbid media. Highlighted on the right are the probes and optics specifically designed for life science applications. The RxN10 probe offers multi-purpose convenience for both liquids and solids analysis, whether in an R&D, benchtop, or pilot scale, as well as single-use manufacturing. The benefit of the RxN10 probe is it accepts a variety of interchangeable optics, whether it's non-contact or immersion, or even single-use, which makes it a highly versatile and easily adaptable instrument in your analysis toolbox. Here, I'm demonstrating the various application and scales that these life science probes can be integrated into. So again, rather than sacrificing on performance and compatibility, we do have dedicated probes for dedicated environments, regardless of the scale. So starting on the left, we're going to highlight the RxN46 non-contact probe, which is optimized to fit the BioPat spectral platform, enabling higher number of experiments to be conducted simultaneously, high throughput systems like the Amber 250 systems, which significantly increases the throughput compared to traditional large-scale bioreactors, and allowing for increased productivity and accelerated development timelines. In the lab scale and pilot scale, we offer a variety of probes that are compatible with this space, one being our single-use optics, which offers a design that has two components, a sleeve and an optic, offering an easy snap-in-place design while delivering reliable, real-time bioprocess measurements with maximum sterility and efficiency. Lastly, on the right is our RxN45 probe, which meets the daunting sampling requirements of compliance, sterilization, port compatibility, and convenience at a manufacturing scale. The RxN45 offers a surface finish suitable for cGMP environments, fits the side ports of large bioreactors, and is compatible with CIP/SIP protocols. Now we will dive into our first application, where the purpose of the study was to demonstrate Raman model robustness across scales. The goal of this study was to address challenges faced when building Raman-based models for monitoring and control at larger bioreactor scales, where running 10 to 20 fed batch experiments at large scale would be costly and time-consuming. Starting at smaller scales like the AMBER system is a powerful tool for addressing the challenges with tighter control over pH, DO, temperature, and feed strategies, ensuring reproducible conditions. This reduces variability in Raman spectra, improving model robustness, and accelerates chemometric model building before scaling up. In this experiment, for the calibration set, an RxN46 probe was used in conjunction with an AMBER 250 analysis module. Raman spectra and offline measurements were collected, and multivariate data analysis techniques were employed to correlate Raman spectra with offline values. The model was then applied to a 10-liter stir tank paired with an RxN10 probe and a biooptic for validation. Both scales maintained the same cell line, product, process conditions, and feeding strategies. This study proved that high-throughput systems provided statistical power for model calibration at a fraction of the cost, but also and ultimately successful transferability of Raman models for glucose and lactate concentration between scales. Now that transferability has been established between various scales of operations, we're going to dive into Raman as a PAT tool for control of critical process parameters. Where traditional methods like offline sampling can be slow, it also can't capture dynamic changes in the bioreactor. Integrating a tool like Raman spectroscopy provides continuous in situ measurements of metabolites without interrupting the process. Let's take glucose as an example. A feedback control loop can be established by integrating Raman data with predictive models. The system can then automatically adjust feed rates to maintain glucose within optimal ranges by establishing a set point with upper and lower bounds for glucose levels. The Raman system scans and outputs glucose values, where the value triggers pumps to adjust the feed and keep glucose within the target range. This prevents overfeeding or underfeeding, which can impact cell health and product quality. Stable glucose levels reduce variability in critical quality attributes such as glycosylation patterns and protein yields. Moving into downstream, the new microflow assembly brings inline Raman measurements to downstream bioprocessing. In contrast to upstream, where processes evolve slowly, compositional changes happen rapidly in downstream. The microflow assembly meets the speed demands by optimizing Raman collection for clear, low-volume samples while allowing liquid material to be analyzed without interrupting the flow. Designed to produce laminar flow within a wide range of volumetric flow rates, pressures, tubing diameters, and fitting options. The flow bench leverages the internal geometries to enhance the total signal-to-noise, which features amplification to deliver 3 to 4 times increased signal over traditional immersion optics, resulting in shorter sampling time, achieving lower limits of detection and finer precision. The flow cell wetted portion, designed for simplicity, cleanability, and consistency, provides flexibility to swap out cells anytime without impacting your process or sterility. Here's a quick look at the substantial signal amplification in the red spectra compared to the traditional methods demonstrated by the green spectra. This flow solution has the ability to generate clean data in some of the most challenging applications. In this application, we're going to start with chromatography because that is the most expensive subset of unit operations. In this case, protein A represents the largest slice within that, being it the single biggest target for innovation. So if we start with the simplest application, which would be column elution, we need to ask if Raman can meet the minimum requirement of monitoring the monomer curve. With the flow assembly, we are able to close the speed gap of the current technique of UV. But not only are we able to close this gap, we are able to do so while also bringing significant improvements over UV methods in specificity. Detection of multiple biomolecules in a single sample is important for applications where multiple analytes need to be quantified in a single step. So the next question might logically be about the ability of Raman to accurately discriminate between the key elements contained. And the answer is yes. Raman spectroscopy has a broader dynamic range than UV absorption, which means that it can detect proteins and other biomolecules at lower concentrations and over a wider concentration range, providing a unique spectral fingerprint that can be used to identify and quantify the protein of interest with high accuracy. This means that Raman spectroscopy can be used to distinguish between the target protein and other contaminants in the sample, leading to more accurate and reliable results, making it a useful and powerful tool for detecting and quantifying low-level impurities. In this last application, we're going to shift gears and showcase how Raman spectroscopy can be a critical PAT tool in pharmaceutical manufacturing. In this workflow, we can demonstrate how Raman monitors in various stages. Starting with that first stage on the top left, reaction monitoring. Raman monitors reaction progress in real time by tracking chemical bond vibrations. It can detect formations of APIs and ensure reaction endpoints are reached without overprocessing. Moving into crystallization, or your polymorph forms, Raman can identify polymorphic forms, so those different crystal structures of the same compound, which is critical as the polymorphs can affect solubility, stability, and bioavailability. In the drying stages, Raman measures residual solvent levels and confirms polymorph integrity during drying. With this, you can prevent incomplete drying or unwanted polymorphic transitions. Moving into the granulation, Raman detects hydrate or solvate formation during wet granulation and maintains correct API form for downstream processing. In the blending stages, Raman checks blend uniformity by mapping API distribution and excipients, which reduces risks of content variability in final dosage. Raman can also be utilized to monitor coating thickness and uniformity in real time, confirming API presence in coated layers if applicable. Tying into the workflow in the previous slide, this slide here highlights the various applications with the design of experiments. In this first one here is API manufacturing, where Raman demonstrated real-time endpoint determination, allowing reaction quenching at optimal ratios. The second here is power blending, where measurements were made on moving powder to ensure homogeneous mixture where a non-contact measurement was established. And the third, tablet coating, where the probe was installed directly into the coating drum to measure coating composition and uniformity. If the topic of Raman spectroscopy interests you, feel free to dive into our application notes available on our website, endris.com. Thank you all for your time.
Thank you, Nicole, for that informative talk. I would now like to introduce our next speaker, Alicia Stell. Dr. Alicia Douglas Stell has worked with CEM for the past 18 years, joining after completing her PhD in analytical chemistry. Her extensive background in chromatographic analysis and her mechanical expertise have been integral in the development of both instrumentation and novel applications. In her role as market development manager, she is continuously exploring new applications and markets for innovative technology focused around sample preparation. Alicia, thank you for joining us today. You have the floor.
Hello, and thank you for that introduction. And thank you to those who are tuning in the audience today. The title of this talk is Fast, Simple, Safe, Master Trace Elemental Testing and Dietary Supplements. So I hope that during the next 20 minutes, I can give you a good perspective on this topic, predominantly coming from the view of sample preparation. Now, dietary supplements, let's take up just a brief moment to introduce them and why we're talking about them, right? And I feel that this is an area that is very consumer driven in our country and globe here in 2025 and forward. And that's predominantly, I think, because we care about these things that are listed on the screen here, right? We want to maintain our health and address any nutrient deficiencies, boost our energy, lower our cholesterol, reduce inflammation, and so forth, right? These are all really good things, right? No one's looking at this list and saying, oh, no, I don't want that, right? And so if there's a way that we can get these benefits through dietary supplements, I think a lot of people are interested, and myself as well, right? I take a multivitamin every single day, and on the days that I don't take it, like say I forgot or whatever, I think I can actually notice a difference in how I'm feeling that day. So that's just my perspective. I'm not here to talk really about the effectiveness of dietary supplements, but just to kind of introduce it as a topic that is of interest in today's world. Now, as noted when I covered the title, we're really going to focus on sample preparation for this talk. And specifically, we're going to be talking about metals analysis. And so what type of sample preparation do you need to do to get metals analysis? That is digestion. So we're going to be talking about digestion, and you need digestion because for analysis to determine the elemental composition of your sample, you need to get that sample into solution, right? And that needs to be a homogeneous solution that we can get a very clear and consistent measurement to determine the elemental content. And so really at the end of the day, specifically if we're looking from the sample preparation perspective, the solution is that solution, right? For us, we need to get a solution of our sample so we can move on to analysis. Now, digestion, how do we achieve that? Well, essentially, that's going to be achieved by heating the sample in an acidic environment. And that can be done via traditional conductive heating, or another option is microwave heating. And we'll be focusing on microwave heating for the solution here today. And here's just a little bit of a graphic to kind of talk about some of the benefits that you gain by using microwave heating. And what we're able to do is kind of heat that sample directly, right? So we can get direct activation of the molecules in that solution. And so the heat is going to get to temperature significantly faster. Also, this is in a sealed environment, right? And so that elevated pressure we achieve is assisting in a faster digestion. But also, it's creating an environment that we're not going to lose those low volatiles or any of our sample. We're not going to go to dryness. And so there's a lot of benefits from that perspective as well. So for the rest of the talk, we'll be focusing on an option of microwave digestion. But before I get into the details of that microwave digestion, I want to talk just a little bit more about the importance of sample preparation. And I thoroughly enjoy getting the opportunity to give the talks that I give because sample preparation is so important and it's often overlooked, right? A lot, obviously, the numbers at the end, right? Your analysis, that's our goal. That's what we're getting to. That's what we need to know. But you can't get there without sample preparation. And a lot of times in a presentation, it's noted what they did. Maybe it's a slide, maybe it's not a slide, right? There's not a lot of time spent often on the sample preparation, but it's so key to the success of the analysis that you're looking for. And so this is a pie graph that was taken out of LCGC magazine that you see here back in October 2023. And what they did was pool a bunch of people and ask them about the importance of sample preparation. And you can see here that it's important, right? We're not going to deny this. So I love the opportunity to sit here and talk about it and talk about what we need, right? And what we need is a streamlined laboratory workflow from start to finish, and that includes sample preparation. And we're going to achieve that by having fast, simple, and safe solutions. And so if you gain anything from the talk that I give here today, I hope it's that you go back and say, okay, that's an option that is fast, safe, and simple for a metals analysis. Okay, and we're going to achieve that on the blade system. And I want to talk a little bit more about what the blade system and how it's going to achieve that. Now, the blade system is a sequential automated digestion system. So you are going to it's running one sample at a time, but then it's automated so that's going to move on to the next sample as soon as that one is completed. It's going to achieve the fastest digestions possible. And it's able to do that by we're running just one sample. So we can focus all of our microwave energy into that one sample and get to temperatures and pressures faster than anything else on the market. It has an integrated camera. I'll take a little bit more time later to talk about that because I think it brings a lot of value to your digestion. It's really easy to use. The safe and simple aspect of this should not be overlooked. It's really needed. And it's flexible, right? Because you're running in sequence, you can have totally different sample types with different acids back to back to back. So you get that flexibility that works well in an R&D environment, but it's also extremely fast. So it covers the high throughput as well. So you get the best of both worlds there. So here's just a quick video to cover a little bit more on that automation that you're going to be able to achieve. So it holds a rack of 24 samples, so you can automate up to 24 at a time before you're moving on and refilling your racks. And what you're going to see here is that when it picks up your sample that's just been digested, it's going to put it back in the rack. So that's your completed sample. But as it moves on to the other one, watch that light, right? So it's going to make sure that you have a sample there that is capped properly. And that's a safety feature, right? That's needed. And then you're going to put it in. You create that environment. We can get the microwave energy, you can get to temperature and pressures quickly and get a fully digested sample. Okay, so the next thing I mentioned was to talk a little bit about that integrated camera, but I also want to bring in the aspect of vortexing. So we'll talk about it when we talk about how you prepare your vessel, but you really do need a stir bar. And the reason you need a stir bar, and you're going to see that depicted here in the camera. So you can always be watching this camera. It's a choice. You can choose to watch what's going on. And that stir bar is going to be, now you see your sample dropped in, is very important because that's going to create this vortexing environment. You see that? And so that vortexing environment that you're creating helps achieve these digestions that are going to be happening in a matter of minutes. But with this camera, you can see the moment that the digestion takes place. And you're going to see it here. It gets kind of dark and then it goes clear. And that's really awesome from a R&D perspective because you can tell when your digestion takes place. So you can know and go back and say, okay, this was the temperature. This was the pressure. I can set up my method to be applicable for exactly what I need in the most efficient manner. So while it's super cool that you can see what's going on, right? It's also really effective and gives valuable information as well. So that's just I think it's a really nice aspect of this system. So now let's discuss the samples that we'll be digesting and the data covered in the presentation here today. So anytime we're doing a study, we're going to want to run SRMs. This is going to help us validate that our methodology is fit for purpose for what we're doing. So in this case, we're going to run applicable SRMs in the dietary supplement arena, which is a multi-element tablet and a ginkgo biloba. As far as the dietary supplements themselves, we're going to run a multivitamin gummy as well as a multivitamin tablet. And now that's really important because those different types of, forms of the supplement matter, right? Gummies are hard to work with. They're hard from a sample preparation perspective. And there is a massive trend right now going towards more gummy supplements. And so it's really important that we have methodologies out there that work for the traditional tablet form, but also for these gummy forms that are becoming really, really popular. And then another thing we're going to look at is some mushroom powders. Also, it's kind of an emerging supplement that I think is getting traction. So we're going to specifically look at some lion's mane powder as well as some reishi powder. So hopefully we've got a nice set of samples here that cover some of the different forms that these supplements would come from, as also some up and coming and interesting dietary supplements as well. So before I get into the actual digestion parameters, I want to talk a little bit about sample weighing and how you're going to get your sample into the vessel itself, right? Because it sounds trivial, right? And pretty much in most presentations, they're going to be like, put your sample in your vessel. But It's not trivial, and it's certainly not trivial when we're talking about elemental analysis, because you want to avoid any sort of contamination from metals that could happen. And so you don't want to use anything that's metallic. You need to be using ceramic and Teflon coated and these options, and they're out there, right? You can see some pictured on the screen here. And so how you're interfacing with that sample, how you're getting that into your vessel really matters. Also, we talked about needing a homogeneous sample, and that's really important as well. It's hard to get a homogeneous sample of a gummy. And so you may need to grind your sample or cut it up into very small pieces. And just think about when you're doing that, that you're again going back to using ceramic or Teflon coated components that are going to not add any contamination, but also help us achieve a homogeneous sample the best that we can. Now let's look at our actual digestion parameters that were used on the Blaine system. So for all the samples, a 0.5 gram was used. And then as far as our acid mixtures are concerned, do take note that when we were using the SRMs and the multivitamins, we did use a mixture that included HF. Now, when we can avoid HF, we're going to. However, in some cases, HF will be necessary. And specifically when running a lot of the SRMs, and in this case, the ones we're looking at here, the methodology that you look at in the certificates will require the use of HF there. And so you need to do that to make sure you're following the appropriate methodology. Now for the mushroom samples, we did not use HF there. So just want to make a note of that. All the samples were taken to the same temperature and then used for the same method forward, which required a 4-minute ramp time and a 4-minute hold time. So look at that, 8 minutes. Your digestion is completed in 8 minutes. Again, fastest available there. For all the data we're looking at, everything was run in triplicate and analyzed on an Agilent 7850 ICP-MS. And I do want to make note that we are following methodology that would be used in the dietary supplement arena across the globe. So that's important as well. So let's look at the data. So as noted, we're going to first start with our SRMs. So we have the multi-element tablet and the ginkgo biloba. And so there's a lot of numbers on the screen here. So let's take a moment here to kind of break them up. Okay, so what we've got is what was detected. And again, this is an average of N equals 3, and all the units you see here are in ppb, so parts per billion. All right, so what was what we actually detected in the sample, the actual concentration of what was expected, and then the percent recovery that you achieved with those. So that's kind of the list going down. And then we had the elements that were qualified for that SRM. We're looking at the data for those. So there were more elements in the multi-element tablet for qualified values than there were in the ginkgo biloba. And the reason we're putting those expected concentrations there, because I want you to note that they quite big range here, right? So we have, as far as cadmium, and where we're at the 80 around 80 ppb. And if we look all the way over to the selenium, we're at 16,000, right? So there's a quite big range here of what potentially could be in these samples that we're looking at. And so that matters, right? What's your dynamic range? What's fit for purpose for your method? And so I just want to show here that we're covering quite a bit range for the methodology that we're looking at here today. You always want to focus on your big 4 heavy metals, so your arsenic, cadmium, mercury, lead. In most cases, those are the ones that are regulated. You need to make sure that you are not above limits for those particular elements. And then we also looked at some of the other elements of interest, so your cobalt, nickel, palladium, and selenium as well. And then so that's specifically why we're looking at these elements, and these are the ones that were qualified for the SRMs, just to qualify that. But overall, percent recoveries look good across the board. So this really gives us the fact that we are like, okay, this method is fit for purpose for what we're trying to achieve. So let's look at the sample results. So again, a lot of numbers, right? Just kind of look at it, you're like, okay, I don't really know what that's looking at. Again, units are in ppb, and this is an average of N equals 3. And here are the actual samples we looked at. So a multivitamin gummy, a multivitamin tablet, and then the 2 mushroom samples, the lion's mane powder and the reishi powder. And again, we're looking at the big four and then some of those other elements of interest. And so what, first off, everything's under the limits. So there's no big red flags here. Okay. So right off the bat, I want you to know we're not concerned as far as any things outside regulation. Okay. Because these numbers, you're like, okay, they'll maybe look large, but they're not, right? We're again parts per billion. So there's no major concerns here. But if you do compare the gummy to the tablet, it looks like maybe we're getting higher values across the board in the tablet, right? Maybe a couple trend of things tend to be a little bit higher in the tablet versus the gummy. And then if we compare the two mushroom powders, we kind of see that maybe there's a little bit more going on in that reishi powder compared to the lion's mane powder. But again, everything was passing here, so no major red flags, but it's always interesting to look at different trends. Personally, I have a child that has a nickel allergy, and this is not just touching her skin nickel, it's in her food, right? So she can't eat any food that has high values of nickel in it. And so it's always interesting to me to look at that. And then I look at the difference between the gummy tablet and the multivitamin tablet. And so I'd be okay, maybe because I'm trying to minimize my daughter's nickel content, maybe that would matter to me. But again, all acceptable across the board. Okay, so in summary, I want to go back kind of to the title, right? Where we're trying to achieve an option for metals analysis that was fast, simple, and safe regarding the sample preparation, the digestion. And I really hope you've seen we were able to achieve that for a variety of different sample types here. Really, it's the fastest you're going to be able to achieve this. It's really simple. It's automated, that removes human error, and there's a lot of different benefits there. And it's safe because this, you're not interfacing with these acids. Your system's taking place with that. So there, I hope you've taken that away. We were able to optimize the workflow where you can reduce the acid, you minimize the prep time, you bring this automation in, you're freeing up your technicians to be running out other tests and so forth. And so really that whole idea of workflow and making that as efficient as possible, I think this fits really well in line with that. And we ran different sample matrices in this case where some had HF and some did not, and we can run it under the same platform. And so that really gives us a lot of versatility and flexibility from our method perspective and what we're able to achieve from sample types on the system. So if you're interested in more information, I would encourage you to go to our website, www.cem.com. What I have listed here, shown here, is our Crazy Dave's periodic table. This is a great resource. It kind of tells all the different elements and gives you the different acids that would be great to digest those elements. But there's tons of other great resources there too, from different application notes, more in-depth samples within this dietary supplement area that we're talking about here. Our other platforms, we talked about our Blade system here. We have a batch microwave system as well. So there's just a lot of information there. So if you're interested, please visit our website. I would not be able to be presenting to you here today if there wasn't a great, amazing team back at CEM doing all of this work. And so they're all pictured here. I think if anybody's worked with CEM, you've seen these faces, you know these people, and you know the passion that they have for what they do. So just a huge thank you to all of them. Again, they're what makes this happen. So that brings me to the end of my presentation, but we want to answer your questions. So if you've got any questions, please reach out directly to me at alicia.stell@cem.com. Or if you want to reach out directly to that team that you just saw pictured, you can reach out to them at elemental.support@cem.com. Either way, we look forward to hearing from you, and thank you for listening today.
Thank you, Alicia, for that informative talk. I would now like to introduce our final speaker of the day, Gerlinda Witte. Gerlinda Witte is the global marketing leader for materials and energy markets at PerkinElmer based in Austria. She has been a chemical engineer and food technologist since 1989 and worked as a sales specialist professional in materials characterization across Europe. Gerlinda, thank you for joining us today. You have the floor.
Hello, everyone. My name is Gelinde Vita and I'm Market Development Manager at PerkinElmer. Thanks a lot for attending my presentation today about mitigating risk while maximizing production in the pharmaceutical industry. PerkinElmer is a global company with more than 5,000 employees worldwide and 1,600 field service engineers out in the field. We have around 20,000 laboratory instruments installed worldwide, accelerating scientific outcomes with cutting-edge technology, world-class service, passion, purpose. That's what we call science for purpose at PerkinElmer. Here, I want to provide an overview about our actual product portfolio for analytical solutions. Starting from chromatography with GC, GCMS solutions, LC and LC-MS solutions, we provide the inorganic spectroscopy instrumentation like AA, ICP-OES and ICP-MS instruments, to molecular spectroscopy systems like IR, UV-Vis, UV-Vis NRR fluorescence instruments, to thermal analysis. When it comes to QA/QC, we offer everything to stay compliant each step of the way, from our analytical instrumentation to our unique one-source service and support. When these tools come together, they work as a system to create safe, quality products you can trust and feel confident about. Good manufacturing practice not only drives the QA/QC inside your company, but it also guides our development of the technologies, tools, and processes we build to ensure you are meeting your regulatory obligations, all while continuously manufacturing compliant products. With that, I will go now more into detail about our solutions for the pharmaceutical industry. Pharmaceutical labs need the flexibility to perform a wide range of analyses, from analytical method development for QA/QC to basic research into new drug candidates to dissolution testing. The Lambda 365 Plus UV-Vis shines in all these applications, and it's 21 CFR Part 11 compliant to meet your most stringent government and industry regulations. Here, I want to provide some information about the quite common application in the pharmaceutical industry, which is UV-Vis analysis of ethanol purity according to pharmacopoeia standards. Ethanol is a key component in pharmaceutical products. It is largely used as solvent, disinfectant, and excipient. In pharmaceutical manufacturing, determination of ethanol purity is a critical quality control measure to ensure drug safety while complying with regulatory standards. The UV-Vis analysis provides a simple and reliable method to determine presence of impurities or adulterants in ethanol. The acceptance criteria are set by the European Pharmacopoeia and the United States Pharmacopoeia. In this application, the Lambda 365 Plus Touch UV-Vis spectrophotometer was used to determine ethanol purity according to the pharmacopoeia standards, with a pass-fail method as a standalone solution that allows for a fast and simple procedure to guarantee high analysis throughput while maintaining accuracy and reliability. A pass-fail method was created in the UV Touch software that controls the instrument. The method parameters are shown here. Furthermore, you can see the results as an absorption spectrum of ethanol. The measured absorbance values for the ethanol samples were all found to be below the USP prescribed limits across the entire spectral range from 235 to 340 nanometer. In the touch workflow procedure, this results in a final green pass message. These results confirm that the tested ethanol could be applied for further pharmaceutical use. Infrared spectroscopy has become a very common tool in the analysis of pharmaceutical raw materials with both mid and near-infrared spectroscopy functioning as commonplace techniques for the investigations of both excipients and active pharmaceutical ingredients. Near-infrared spectroscopy is the most common spectroscopic technique for the interrogation of incoming raw materials. Mid-infrared spectroscopy generally contains the most information regarding the chemical structure of an analyte, making it particularly useful for identification and analysis of active materials in pharmaceutical samples. The PerkinElmer Spectrum 3 3-range infrared spectrometer provides a high-performance solution for pharmaceutical analysis across the infrared spectrum. From routine identification and verification to advanced research applications, you need the performance and flexibility to quickly, confidently, and cost-effectively analyze a wide range of samples. The Spectrum 3 FTIR spectrometer provides the sampling flexibility and performance in mid, near, and far infrared ranges through a single instrument to advance research and new product development in pharmaceuticals. The highly configurable platform provides dependably consistent and trouble-free operation. It is a configurable multi-range FTIR system and can be upgraded with microscopy, imaging, and hyphenation. Moving on in my presentation, I want to provide some insights in the analysis of raw materials using the Spectrum 3 dual-range spectrometer. We describe how the Spectrum 3, together with our 21 CFR Part 11 compliant Spectrum IR software, provides a high-performance solution for mid and near-infrared analysis of materials used in the pharmaceutical industry. Near-infrared spectroscopy is one of the most common spectroscopic techniques for the interrogation of incoming raw materials. This is due to the robust nature of the technique and simple sampling methods, which allow for quick and simple data collections. There are two main sampling accessories which may be used to investigate pharmaceutical raw materials in the near-infrared region. One of them is our near-infrared sampling module and the other one our remote sampling module. One particularly useful application of near-infrared reflectance spectroscopy is the discrimination between spectroscopically similar materials with the aid of principal component analysis. An example of this is the discrimination between different grades of avicel and microcrystalline cellulose. In the picture on the left side above, you can see that we have collected near-infrared spectra of 7 different grades of avicel. In this scenario, then we use Simca modeling, which is incredibly useful for the differentiation between grades of avicel. The model was able to recognize 100% of the samples with each grade correctly. On the other hand, mid-infrared spectroscopy generally contains the most information regarding the chemical structure of an analyte. Therefore, it is particularly useful for the identification and analysis of active materials in pharmaceutical samples. The search function in Spectrum IR may be used to identify APIs by comparing the sample spectra against reference spectra in a library. We took spectra of both the APIs and packaging materials, measuring using the Spectrum 3 with universal attenuated total reflectance accessory, which we call UATR. You can see the overlaid spectra here. As I already mentioned, the Spectrum 3 is not only expandable in the wavelength ranges, it can be also upgraded to a microscope or imaging system. With our new Spotlight Aurora microscopy and imaging system, we offer the best-in-class visible imaging system, and our stage allows for measuring of non-flat samples. Our Spectrum IR software, which we also offer for our Spectrum 3, provides assisted workflows. That means that all sample workflows can be automated for all users of any skill levels. Furthermore, we offer multi-detector options, which corresponds with the detector options on the Spectrum 3. This offers ultimate inflexibility. And then we are able, with our Spotlight Aurora microscopy and imaging system, to tackle the most demanding applications with research level macro and micro infrared sensitivity and performance. One example for application using FTIR imaging is the identification of counterfeit pharmaceuticals. Counterfeiting of pharmaceuticals and the proliferation of substandard drugs constitute a serious health risk for the world population, including both industrialized and developing countries. The manufacturing, sale, and distribution of counterfeit drugs are not only serious crimes, but also lead to failed treatment, disability, and even death. Loss of revenue for the pharmaceutical industry and potential exposure to huge damage claims will push up the price of legitimate pharmaceutical products. First of all, we took near-infrared spectra using the Spectrum 3 NIRA, and where you see the green spectrum is the genuine product and the red one the counterfeit. Then we created band ratio images for both products. The left one is the genuine product and the right one the counterfeit. The distribution of caffeine in this case is clearly homogeneous in the genuine product, but very localized in the counterfeit product, which reflects different manufacturing processes. So the Spotlight Aurora imaging is ideally suited to the rapid identification of counterfeit drugs based on the distribution of ingredients. As previously mentioned, the Spotlight Aurora offers the new and unique C-stacking options, which provide the opportunity to measure curved samples without any sample preparation. Curved samples made stitching together in high-definition imaging was problematic, as you see above here. Different regions have different heights even after cutting. The picture below shows that the C-stack focus optimization is fixing this problem. And also behind every single point, there is the infrared information stored. With that, I want to move over to our inorganic spectroscopy product portfolio. The International Conference of Harmonization of Technical Requirements for Registration of Pharmaceutical Products for Human Use, the ICH, has completed its guideline for elemental impurities. This is known as ICH Q3D. The guideline provides a global policy on limiting metal impurities in drug products and their ingredients. The analytical tools are outlined in Chapter 233, which are ICP-OAS and ICP-MS. Also, the use of a closed vessel sample preparation technique, such as microwave digestion, is required. PerkinElmer offers a complete integrated solution to ICH Q3D and USB 232 and 233, which includes fast, safe, and cost-effective sample preparation equipment, intelligent sample handling that includes automated auto-dilution systems for elemental impurities method, choice of the best-in-class ICP-MS and ICP-OS, including software solutions, complete validation services, enhanced security software to help comply with 21 CFR Part 11. Our integrated USB compliant solutions provide calibration and check standards for the analysis of impurities in pharmaceutical products and raw materials. Our pre-mixed multi-element standards can be used to calibrate for each class and administration route as defined by the ICH, including the big four: arsenic, cadmium, lead, and mercury. We have the method templates, expertise, and technology to provide your laboratory staff with the tools and knowledge they need to precisely measure levels of metals in pharmaceutical products at the limits defined by ICH and USP guidelines. Finally, moving on to chromatography. Our GC 2400 platform provides smart connectivity, simplified operations, and sustainable design. Furthermore, it provides efficient workflow solutions for gas chromatography. The GC 2400 platform is backed by a portfolio of separation chemistries and solutions that offers labs an efficient workflows experience. Analytical labs are faced with growing demands for increased throughput, improved operations, and more efficient processes. Software plays a crucial role in this area, helping automate chromatographic workflows through integrated data collections, analysis, and reporting. Our Simplicity Chrome chromatography data system software, which is working with all our chromatography systems, is an easy-to-learn, modern, and intuitive CDS software platform, which enables efficient control of our PerkinElmer HPLC, UHPLC, GC, and GCMS solutions while integrating the overall chromatographic workflow. With that, moving on to an important application in the pharma industry, which is USP 467, the analysis of residual solvents. Organic solvents are commonly used in the production of APIs and pharmaceutical products. The solvents are used for varying purposes, including as synthesis reaction media in the separation and purification of API during synthesis. The manufacturing process does not always consume or break down all of the solvent. Therefore, the residual solvent levels must meet health-based limits to ensure patient safety. USP 467 identifies three classes of residual solvents, each distinguished by their own or potential health risk for patient safety in pharmaceutical. USP 467 identifies gas chromatography with flame ionization detections as the preferred analytical technique for analysis of residual solvents in pharmaceuticals. Due to the prescribed GC method settings from USP 467, it has a long runtime per sample, which can slow down the release of finished pharmaceutical products for manufacturing. The long runtime can lower the number of samples that can be analyzed daily. To overcome the time limitation and increase sample throughput, improvements to the method have been made by optimizing the analytical parameters. These improvements decrease analysis time for residual solvents, allow faster QC analysis, and increase sample output, and this is what is shown here in this example. Our actual LC portfolio consists of the LC 300 HPLC and UHPLC systems. They help you to get up and run faster and easier with automatic priming and purging. They improve productivity and minimize error with prep capabilities through automation of tasks. They maximize uptime through intelligent maintenance notification and routines. And our patented PDA and integrated easy-to-use spectral processing tools provide market-leading performance for the most challenging laboratory applications. Furthermore, I want to introduce our QSight 500 LC-MS system. The QSight 500 excels in small molecule contaminant analysis, handling the toughest samples without sensitivity loss or incurring cleaning downtime. A simplified blend and eject sample preparation approach frees up time and money compared to the typically used squashers method. The QSight 500 has leading class quality in handling of the toughest samples at less cost with less time while growing capacity. I just want to provide a short insight into the determination of nitrosamine impurities in APIs using our QSight LC-MS system. Nitrosamines are highly potent mutagenic impurities formed from secondary or tertiary amines under acidic conditions. Regulatory agencies, including the US FDA and EMA, have imposed strict limits on nitrosamine levels in drug substances such as sartans and metformin following their discovery in 2018. To ensure product safety and regulatory compliance, trace level detection down to 0.1 ppb is required and is achievable through sensitive LC-MS workflow. The QSight LC-MS delivers fast, selective, and robust quantification of nitrosamine impurities in APIs, meeting global regulatory standards for safety and quality, ensures compliant manufacturing and peace of mind in pharmaceutical QC workflows. With that, I want to come to an end of today's presentation. As I have shown, PerkinElmer is a reliable partner for the pharmaceutical industry by offering a complete solution of analytical instruments, software which is full pharma compliant, including validation service and support. Thanks a lot for your time and listening.
Thank you, Gerlinda, for that excellent talk, and thank you to all the other speakers that participated in today's event. I want to thank the audience for attending and for participating in the afternoon session of this virtual symposium, Emerging Trends in Pharmaceutical and Biopharmaceutical Analysis, presented by Spectroscopy, the Society for Applied Spectroscopy, and the Koblenz Society. Thank you again to our sponsors, CEM, PerkinElmer, Thermo Fisher, Metrome, and Endress & Hauser. We want to remind you to take advantage of the resources from those sponsors, which you can find on the bottom of your screen. If you would like a certificate of attendance for this event, please send an email within a week of the event to training@sas.org. That's training@sas.org. You will receive an email alerting you when this virtual symposium will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. We hope to see you all tomorrow for day 2 of Emerging Trends in Pharmaceutical and Biopharmaceutical Analysis. Goodbye.